Hi everyone, my name is Natália Koch and I'm a postdoc researcher at the University of Minnesota, United States. I'm going to present my work entitled Photobiont genus and carbon concentrated mechanisms matter for lichen ecology. This work is co-authored by James Landmer, Christy McCain, Aaron Manzito-Tripp, and Daniel Stanton. So lichen photobionts are influenced by environmental changes and they may limit the presence of certain lichen species in unfavorable habitats. One example is Trentepohlia and their relation to humid and warm environments, which may limit the presence of certain lichen species that are associated with them, with this genus. Carbon concentrated mechanisms that I'm going to call here CCMs are a widespread phenomenon evolved to increase the ratio of carbon and oxygen within the chloroplast. The evolution of this mechanism in vascular plants was a major innovation, but some algae have lost this mechanism, while others have kept this. One example of algae that kept this mechanism is the genus Asterochlores, and I show here inside the cell the pyrenoid, which is the structure that concentrates carbon. This gain and loss of carbon concentrated mechanisms is associated with significant environmental range shifts, both for plants and algae. And algae, we can mention, for example, the shifts from land to water and from water to land. Examples of CCMs are in plants, in green algae and cyanobacteria. In green algae are the pyrenoids, in cyanobacteria the carboxysomes, and in plants the C4 and CAM photosynthesis type. Lichen photobionts with this mechanism might be more advantaged when they are in low CO2 concentration, which is the case of thalai with higher water saturation within that. So we have the question, do carbon concentrated mechanisms constitute a major trait in structuring lichen communities? To answer this question, we used a large-scale data set of lichen communities from Eastern North America that was sampled by expert lichenologists and led by Dr. James Landmer, Dr. Richard Harris, Dr. Christy McCain, and Dr. Aaron Manzito-Tripp. The complete dataset comprises 1,208 species, and it represents the total lichen biodiversity, which means all substrates and growth forms that were inventoried in 630 sites. The photobiont genera were retrieved from publications and classified based on the presence and absence of CCMs, which is shown here in the table. So green absent genera, green present genera, cyanopresent, they are all present genera as well. So that's the total genera that we had. And we looked for publications that had phylogenetic information on it. For the environmental gradient, we retrieved data from RoadClim, and to show here at this presentation, I'm using mean annual temperature and precipitation. To explain the results, I'm first going to show an example of the graphs that I am presenting, and that are here. In the y-axis, I'm showing precipitation, in the x-axis, temperature, and the dots are all the sites that were sampled, and the color-coded is based on the proportion of species with carbon-concentrated mechanisms, the ones that I'm showing separated by the trait category, and relative to the total pool of species. So I will show all the 603 sites based on this proportion. Higher proportion are shown in warmer colors and lower proportion in colder colors. So higher in red and lower in blue. So summarizing our main results, we are showing here the green algae without pyrenoids, the green algae with pyrenoids, and the cyanobacteria genera. So based on the proportion, as I mentioned, we already, just by looking at these graphs, we can see that the patterns were different comparing these groups. So we used generalized linear models to test the relations, And we found that precipitation and temperature were significantly related to all these groups, with the interaction being also significant when we looked at cyanobacteria. So green algae without pyrenoids had higher proportions in sites with higher temperature and intermediate precipitation, while green algae with pyrenoids had higher proportions at sites with intermediate mean annual temperature and higher annual precipitation. And cyanobacteria had higher proportions in intermediate temperature and higher precipitation, with the lower proportions in all over the areas and with significant interaction. Highlighting here, we show that the green algae without pyrenoids that had the highest relation to what we saw is the genus Trentepohlia and Symbioclaris, that both had similar patterns when we compared these proportions and when we compared the temperature and precipitation variables. So we highlight that some chlorococcoid photobionts were more similar to other non-chlorococcoid, which means that CCM lacking photobionts were similar to each other, which here we show Trentepohlia, which is a non-chlorococcoid, and Symbioclaris, that is a chlorococcoid photobiont. So patterns in cyanolichens different from CCM containing green algae, and in some ways they were more similar to photobionts that lack CCMs. In conclusion, we can say that presence absence of CCM seem to be in part determining green photobiont distribution, which means showing here by temperature and precipitation, analogous to photosynthetic mode in vascular plants. But further investigation is needed, and to continue this work, we aim to, as next step, to test the phylogenetic signal of photobionts and microbionts. I would like to thank for this work our researchers involved in data collection, identification, and the database creation, and for funding the NSF Dimensions of Biodiversity, NSF Division of Environmental Biology, and the Western Pennsylvania Conservancy for funding. So thank you so much, and I'm open for questions.